Bismillahirrahmanirrahim. Our today's lecture topic is microscopic structure of spleen, thymus, GALT and MALT. Our objective for this lecture will be microscopic features of the spleen and microscopic features of thymus, and we will define the terms GALT and MALT. So we start with the spleen. In the previous lecture we have discussed the location of the spleen, that it is present in the abdomen in the left hypochondrium just behind the stomach, and it is the main organ where the filtration of blood is taking place. And our spleen contains large quantity of lymphoid tissue, and it is the only lymphoid organ which filters the blood, a very important organ in defense against blood-borne antigens. It is also a site of old erythrocyte destruction. It produces antibodies and activated lymphocytes. If we see the microscopic structure of the spleen, it is filled with reticular tissue containing reticular cells, reticular fibers, lymphocytes and macrophages, and it is an encapsulated structure. As in the previous lecture we have discussed that it is a encapsulated, completely covered by capsule, and it is a type of secondary lymphoid organ. So the capsule is covering completely spleen, and from its inner side it extends or sends septa inside, which we name as trabeculae. These trabeculae contains trabecular arteries and veins and a little bit support to the splenic tissue, but the main infrastructure of the spleen is found by reticular fibers and cells. We divide the spleen or this microscopic structure of the spleen into two parts known as white pulp, which constitute almost 20% of the spleen, and the rest is known as red or splenic pulp. So we have two parts of the spleen structure, white pulp and red or splenic pulp. Now, like we have talked about lymph node, that it is a peripheral cortex and central medulla. We don't have cortex and medulla here. We have white pulp and red pulp. Now white pulp means that there are two structures which combine together, and we name that area as white pulp. That is lymphatic nodules with the germinal center. The same we have discussed in the lymph node. Along with that we have periarterial lymphoid sheath. So these two structures, lymphatic nodules and periarterial lymphoid sheath, they combine together and we name this as white pulp of the spleen. Now this lymphatic nodule is easily differentiated from lymphatic nodule, that in the mass of the lymphatic nodule an arteriole is passing, which is known as central artery. So it is one of the identifying feature of lymphatic nodule of the spleen, that inside that lymphatic nodule a central artery is passing, and we will not find any central artery or structure like that in the lymphatic nodule of lymph node. So later on we will see in the slide also. So white pulp means lymphatic nodule and periarterial lymphoid sheath, and the red pulp, which is also known as splenic pulp, it contains pulp arteries, venous sinuses, also known as splenic sinuses, and splenic cords. These splenic cords are comprised of lymphocytes, and we name also them as cords of Bell-Roth. So red pulp and white pulp. Now if we see the structure of the spleen, we have a capsule as we have discussed. This is a capsule. From capsules trabeculae are invaginating inside into the substance of the spleen, containing trabecular arteries and venules with them. So dividing the structure into portions. And the other thing which we see lymphatic nodules which are scattered. They are not specifically in one place like in lymph node they are on the peripheral side, but here they are scattered. So that part of the spleen where lymphatic nodule is present, that specific part is known as white pulp, and the rest of the part of the spleen is known as red pulp. So here we can see the lymphatic nodules forming the white pulp. So we cannot specify the location of the white pulp because these lymphatic nodules are scattered in the whole structure of the spleen, and along with the lymphatic nodules we can see the same area in the center is germinal center, which contains B lymphocytes, and a peripheral zone which contains small lymphocytes and darker in color, the peripheral zone known as mantle we have talked about in lymph node. And if you appreciate, each lymphatic nodule has a central artery which is passing through it. So in every lymphatic nodule we can appreciate the central artery. Now one of the important thing we will discuss again in the later slides, that it is known as central artery not because it is passing through the center of the lymphatic nodule. It is eccentric. It is passing not from the center but away from the center. So place can be anywhere near to the center or maybe toward the periphery. So this is known as eccentric position. So central artery passes through the lymphatic nodule on eccentric portion. The reason we will discuss in few minutes. And that part is known as white pulp. The rest of the area is known as red pulp. The red pulp contains splenic cords in the red pulp. Now what are these cords? The same way we have talked about in lymph node that we have medullary cords, which are actually the lymphocytes arranged in shape of cords. In the same way here the lymphocytes are arranged in shape of cords and we name them as splenic cords, or also known as cords of Billroth, and in between them we have spaces which are known as venous sinuses or splenic spaces. Along with these we can see the trabeculae, and with the trabeculae we can see arteries and venules present in the trabeculae. Also in the red pulp we see some arteries which are named as pulp arteries, which are the constituents of red pulp. So this is about the splenic structure. We have a capsule, we have a trabeculae extending down containing trabecular arteries and veins. We have white pulp which comprised of the lymphatic nodules having in the center, germinal center, a lightly stained area, a peripheral zone containing the darker stained area, along with the central artery in the periphery. Now if we talk about the larger or magnifying view of the spleen, we can see this is a white pulp area containing germinal center, peripheral zone, and a central artery in the lymphatic nodule forming the white pulp of the spleen, and rest of the area is the red pulp along with the trabeculae showing the trabecular vein inside. And here we can see the purplish cells which are very smaller cells in size. They are the splenic cords, or also known as venous cords or cords of Billroth, and in between the spaces we have are the venous sinuses which are separating the splenic cords. Now we look at the schematic diagram of the red pulp. What we can see splenic cords. So this all bluish coloration structures are showing the splenic cords, and these pinkish areas are showing the splenic sinusoids, and in between them these red are the red blood cells which are passing through with the other cells. So if in a very magnifying view we can see red blood cells arranged, that we have sinusoids and splenic cords, which are the collection of lymphocytes. And these sinusoids are, if you can see, they are connected and enclosed by fine reticular fibers which are acting as a supporting tissue to that structure. And we can see that red blood cells from the area which is known as splenic cords, they are passing into and out to the sinusoids. Now this is a diagram which is showing open and closed circulation in the spleen. So actually the spleen is supplied by splenic artery. When it reaches to the hilum, which is the opening in the spleen, it divides into branches, and these branches are known as trabecular arteries. So through the trabeculae at different places they enter into the splenic structure, and the trabecular arteries enter the parenchyma as arterioles. These arterioles which are arising from the trabecular artery, we name them as central artery. So splenic artery divides into trabecular artery, and trabecular artery give rise to central artery. Now a very important thing which is mentioned here is that central artery is covered on its side by a specific type of cell which are T lymphocytes, and we name them as POLs, that is known as periarteriolar lymphoid sheath. So these cells are completely covering the central artery. As it penetrates into the parenchyma, it meets with a lymphatic nodule, as we have seen in the previous slide that central artery is passing through the lymphatic nodule. So as the central artery is passing through the lymphatic nodule, the germinal center is a little bit on the side, and here B lymphocytes or lymphoblastic cells are formed and present there, and the central artery is passing through that lymph. So when we say white pulp, we are talking about these two structures. We are talking about the lymphatic nodule with germinal center, and we are talking about the central artery covered with periarterial lymphatic sheath. They contain T cells along with macrophages and plasma cells forming the periarterial sheaths. Now these central arteries plus lymphatic nodule is a white pulp, as we have discussed. In the developing lymphatic nodules, because of the rapid proliferation of lymphoblasts, these lymphoblasts push the central artery into the eccentric position. That is the main reason that why central artery are passing not from the center of the lymphatic nodule but from the side, that the active proliferation of lymphoblasts, they push the central artery aside, not in the center but away from the center in any place which is known as eccentric position, but still the artery is named as central artery. So not due to its position. After that, each central artery which is passing through the lymphatic nodule, it leaves the white pulp. This is the white pulp area. So as it is leaving the white pulp, it enters into the red pulp. So the same central artery which is coming from the trabecular artery, now from lymphatic nodule enter into the red pulp. And when it enter into the red pulp, you can see that it is lacking the periarterial lymphatic sheath. There is no periarterial lymphatic sheath in the red pulp area, and when it enter into the red pulp area, that artery is known as pencillar arterioles. And these pencillar arterioles, if you can see here, ultimately continuous as capillaries or connect with the capillaries. So we have here two concepts of circulation in the spleen: an open circulation and closed circulation. What does it mean? So from trabecular artery the blood comes to central artery, from central artery it enters into red pulp, from red pulp central arteriole it becomes peninsular arteriole, and that peninsular arteriole in some part of the spleen they are connected to the sinusoid completely. So the blood is passing from arteriole to the sinusoid directly. There is no gap in between them. This type of circulation is known as closed circulation, which is a common circulation in the human body, that arterioles are entering into capillaries or draining into sinusoids. So if there is a direct connection between a peninsular arteriole and sinusoid, we name it as closed type of circulation. On the other hand, if you can see the same peninsular arteriole on the other side, it is not entering into sinusoid, it's just entering into the red pulp area where we have splenic sinuses and splenic cords. So they are releasing their material into the splenic cords, and from there the blood re-enter into sinusoid. So there is a gap present here. That's why this gap is known as type of open circulation. So we have two types of circulation in the spleen: closed circulation, where periarterial peninsular arterioles are connected to sinusoid directly without any gap, and on other side peninsular arterioles are entering into the red pulp area between the splenic cords, and then the blood or material re-enter into the sinusoid. So this type of circulation is known as open circulation. This red pulp structure, which contains the B lymphocytes and T lymphocytes, again as we have seen in the previous picture, they are covered by reticular fibers and supported by reticular fibers, and also contains macrophages, other leukocytes and RBCs. And these sinusoids are lined by specialized cells which are known as staph cells. So we can say that the blood flow in the red pulp has two destinations. One is known as closed circulation and other is known as open circulation. Now this is a diagram which is actually showing the arteriole, a venule, and in between a sinusoid, and the other structure which are present are the splenic cords, which contains lymphocytes, B lymphocytes and T lymphocytes. So this diagram is mainly showing us the red pulp area, the arteriole which is coming towards the sinusoid, showing a closed type of circulation, that it directly connected with the sinusoid, and on the other side they are opening into the splenic cords, and from splenic cords, from the free area over here, the RBCs are re-entering, or the cells are re-entering into the sinusoid. So this type of circulation is known as open circulation. Now the sinusoids of the spleen, they lined by a specialized type of elongated endothelial cells, and they are named as staph cells. These cells, they are elongated cells and we name them staph cells, and they are covered or supported by a fine network of reticular fibers, which not only supporting the sinusoid but it is also supporting the splenic cords. Another very important function of these staph cells, that they allow only the red blood cells which are normal in function and normal in size, so they can penetrate inside and pass through the venules, while those RBCs which are swollen up or which has completed their life, or we can say the old RBCs, they are not allowed to enter into the sinusoid. So these staph cells are preventing these old RBCs or damaged RBCs to re-enter into the circulation. And here, as we have discussed, there is splenic cords, in between them we have macrophages. So those macrophages are responsible for phagocytosing these swollen or old RBCs. So this is all about the spleen. So just we will repeat finally the structure of a spleen, that we have it is an encapsulated structure covered by capsule, and from the capsule trabeculae are penetrating inside, containing trabecular venules and arterioles and branches of the splenic artery, and it comprised of white pulp and red pulp. White pulp is formed by the lymphatic nodules, which are centrally placed germinal center, a peripheral dark stained zone with small lymphocytes, and a central artery which is eccentric in position in each lymphatic nodule, along with periarterial lymphoid sheath, which we can't see in this diagram. So this is the white pulp, and the rest is the red pulp, which is containing splenic cords and venous sinuses, and the pulp arteries already we have discussed. So this is all about the spleen. Our second topic is thymus. Already in the previous lecture we talked about its position, that thymus is present in the lower part of the neck or in the upper part of the chest in the central area, which is known as mediastinum. What is mediastinum? Mediastinum is a central septum present in the thorax between the two lungs. So it lies anteriorly in the thorax, in the upper part, in the mediastinum, and as we can see, this diagram is showing the thymus of a child, because it is a primary lymphoid organ, so it is very much functional in childhood, and as the puberty occurs or a person become adult, this tissue reduces or regresses, and its function is taken up by the spleen, lymph nodes, and other lymphatic tissues. So logically we can see that the thymus over here is showing a lot of cells over here, but if we compare with the adult thymus, we can see only some bits of these cells are present there, but rest of the whole thymic area is changed into areolar tissue, which is a type of connective tissue. Thymus is also a encapsulated structure and primary lymphoid organ, and it is covered by capsule the same way the capsule is penetrating inside and forming trabeculae. So these trabeculae are doing a very important thing, that they are dividing the inside of the thymus into incomplete lobules. That's why when we see the lobule or thymus grossly, it is a lobulated appearance structure. It is a bilobed structure, and the trabeculae extends inwards to form lobules. Now each lobule consists of a dark stained outer cortex and light staining inner medulla. So as we have discussed in the lymph node has only one cortex and central medulla, but in thymus, for example, if we have 10 lobules, so we will have 10 cortex and 10 inner medulla. So that's how it differentiated from the lymph node. The cortex contains densely packed lymphocytes, but there are no lymphatic nodules. So that is one of the differentiating point of thymus with the lymph node, that the thymus contains no lymphatic nodules, but the lymphocytes are densely packed in shape of cortex and medulla. The medulla contains fewer lymphocytes but more epithelial reticular cells. Medulla also contains specialized thymic corpuscles known as Hazel capsules, and these thymic corpuscles are oval structure consisting of round walls of flattened epithelial cells. So we see the picture and then we discuss about them. So small lymphocytes are present in the cortex and large lymphocytes are present in the medulla, which are lymphoblasts. So this is the diagram which is showing the thymus. So the outer part is the capsule. So capsule is sending the septa, and septa are penetrating deep and dividing this whole tissue into lobules. So many lobules we can appreciate, and each lobule has a darkly stained peripheral zone and a lightly stained central zone. So this darkly stained peripheral zone is known as cortex, and lightly stained central zone is known as medulla. So now the cortex and medulla are so many the lobules are. So it depends upon how many lobules are present, that many cortex and medulla will be present, which is one of a very important differentiating point. The cortex of the thymus is densely populated by T lymphoblasts, and these T lymphoblasts are known as thymocytes. So the major cells of the thymus, which are the T lymphocytes, we name them as thymocytes, which are present in the cortex, also present in the medulla, but mainly they are present in the cortical region. Now before we go to the cortex, these trabeculae contains not only blood vessels, but they also contains adipose tissue or adipose cells. As we have discussed that in fetal life there is this is fully functional structure, but as it grows and puberty occurs, this adipose tissue taken up most of the part of the thymus. So when we say the cortex contains thymocytes. Along with that, they contain specialized cells which are known as thymic epithelial cells. Generally, we name them TEC cells, thymic epithelial cells, and they act basically as epithelial and reticular cells, and they are connected together by desmosomes and occluding junctions. So they are not only providing structural support, but we divide them into three types. So the first type of specialized thymic cells are known as squamous T cells, which are responsible for blood thymus barrier. Over here, they are mainly present in the area where the trabecular are passing and the capsule. So at the peripheral zone of the thymic cortex, the cells are present, which are squamous T cells, and they form a blood thymus barrier. The second type, which is present in between, are known as stellate T cells. They form a cytoreticular structure where macrophages and developing lymphocytes are attached. So over here in the mostly central zone, stellate T cells, thymic epithelial cells are present, and they provide support to the macrophages as well as lymphocytes. And these thymic epithelial cells act as a antigen presenting cells. The third type is known as squamous cortical cells. They form sheet like structures present there, and they are the structures which are present on the more inner side of the cortex, and they form a barrier between the cortex and medulla. So three type of thymus epithelial cells are present: squamous T cells in the peripheral zone, which are forming a blood thymus barrier near the trabecular capsule. In the middle zone, we have stellate T cells, which form cytoreticulum supporting macrophages as well as lymphocytes and presenting antigen presenting cells. And the third type, which are present near the medullary part, they are again squamous cells, but they present in shape of sheaths, and they act as a corticomedullary barrier. If we go to the thymic medulla, mature lymphocytes are present here, and medullary thymic T cells are also present over here. They also make a boundary wall or a barrier between the cortex and the medulla, and they also act as a cytoreticular tissue for supporting the different cells. Now, in the medulla, there is a one of a major identifying feature is presence of thymic or Hazel capsules, which are large aggregates of thymic epithelial cells. They are flattened epithelial cells and they appear in shape of whorls and whorls. In next slide, I will show you. And their main function is that they secrete cytokines. Thymus is mainly the site of T lymphocyte differentiation, and in term or in result it induces self tolerance and help in our immunity. This diagram is showing cortex of the thymus, so we can see lymphocytes are present, and in between them these are larger cells which are Tech cells or thymic epithelial cells present there. If we move to the medulla, we have smaller number of cells are lymphocytes, and the large cells are known as Hazel capsules, which are secreting cytokines, and they are large epithelioid cells, and we identify them by their presence that they are whorls of epithelioid flattened cells. Here is the diagram which is showing a cortical zone of a active thymus, in which we can see lymphoblasts densely packed together, and they are connected together by desmosomes. You can appreciate this, so they are supporting them, and along with that we can see the thymic epithelial cells or Tech cells which are present in the cortex area and forming the barriers and the support to the underlying tissue. This is a magnified area which is showing the medullary part and the cortical part. So here we can see the cortex with thymic lymphocytes are present there. This is the trabeculae containing the blood vessels, venules, arterioles and adipose tissue. And here we can see the degenerative centers of thymic Hazer capsules. So they are flattened epithelial cells and they are formed in whorls and whorls or round structures forming a large size of cells known as Hazel capsules or thymic capsules. And in between them intermingled cells are epithelial reticle cells, which are the major supporting cells, along with the macrophages also present there. So that is about the thymus. Now there are two terms we use in lymphoid system. One is known as MALT. MALT means mucosa associated lymphoid tissue, and GALT means gut associated lymphoid tissue. So GALT is actually the part of MALT. Mucosa associated lymphoid tissue means that in mucosa of the different systems we have previously discussed, like gastrointestinal tract, respiratory tract, urinary tract and reproductive tract. These are the tracts which are directly in relation with the pathogens. Most of the lymphocytes are activated by antigen presentation, include lymphocytes, spleen, and they comprise of also MALT. So gastrointestinal tract, respiratory tract, genitourinary sacs are common site of invasions of pathogens because their lumen is directly in contact with the exterior of the body. So to protect from the pathogens, the mucosal surface of these systems act as a barrier and first line defense tissue. This mucosa contains large and diffuse masses of lymphoid tissue containing lymphocytes, plasma cells, antigen presenting cells, and also the lymphatic nodules, and collectively we name them as MALT. And if we go to the GALT, GALT is actually gut associated lymphoid tissue, which is a part of MALT. So we can say that 70% of body immune cells are present in the mucosa which contains lymphoid tissue. So 70% of immune cells are present in the MALT and rest are present in the other tissues. So when we talk about the GALT or gut associated lymphoid tissue, we use the term mucosa. Now what is mucosa? First of all, we will talk about this. Is a lumen structure, a tube of our gastrointestinal tract. It has four layers. Mucosa is the most inner lining. Then after the mucosa we have submucosal layer. After submucosa we have a muscular layer which is known as muscularis externa, and the last layer is known as serosa adventitia, which is a connective tissue and epithelial layer. We will talk inshallah in gastrointestinal tract in detail. Here our main concern is mucosa. That what is mucosa actually? So when we see the wall of the lumen or gut tissue, what we see, the most inner lining is known as mucosa. Or when we talk about a common example, that if we see our lip, the most outer part of the lip is skin, and inner side, which is a wet and lubricated through saliva, that part is known as mucosa. So mucosa is basically comprised of three layers. The most inner layer is epithelium. In the epithelium lecture we have talked about that epithelium is covering the body from outside as well as inside of the body. So outside means we are talking about the epidermis, the skin, and inside means this luminal structure. So this lumen is lined by epithelium. So in different places we have different epithelium, but mainly the gastrointestinal epithelium is simple columnar epithelium. So just below the epithelium we have a very fine layer of connective tissue which is known as lamina propria. So lamina propria is a second layer inside the epithelium forming the mucosa containing connective tissue, and below that we have a very fine smooth muscle layer which is known as muscularis mucosa, which is mainly responsible. The contraction of this layer causes the secretions to go outside the tissue into the lumen. So mucosa comprised of epithelium, lamina propria and muscularis mucosa. So most of the gut associated lymphoid tissue or gall tissue are present in the lamina propria. They can be present in the second layer which is known as submucosa, which is present below the mucosa. So that is the meaning of mucosa. So most of the GIT has a similar mucosa with different epithelial and lamina propria concentration and types. So here is an example of GALT, the ileum, which is a part of intestine. If we can see, we have epithelium, which is simple columnar epithelium. These projections are known as villi. And if we can see, below that we have lamina propria, and below lamina propria this portion is known as submucosa, submucosa, and this portion is known as lamina propria. So if we can see, the lamina propria is filled with lymphatic nodules and diffuse tissue is also present there. So over here in the ileum, these lymphatic tissue is densely packed. In the whole intestine it is present, but in the ileum it is a characteristic feature of ileum that the lamina propria contains the densely packed lymphoid tissue, and we name it as Peyer's patches. So the Peyer's patches is a type of GALT, gut associated lymphoid tissue, and it is actually lymphatic nodules in the ileum, but we name them as Peyer's patches. They can be mostly in the nodular shape, but they can be diffused. This is one of the example of GALT, that is Peyer's patches. Another example is our appendix, which is present just below the cecum, the large intestine. So if you can see here, this is the lumen and epithelial lining of the appendix, and then as we see the lamina propria, we can see the diffuse lymphoid tissue is present, as well as the lymphatic nodules are also present in the lamina propria. So in the mucosal portion, epithelium, lamina propria containing lymphatic nodules with the same germinal center in peripheral dark stain zone, along with some places you can see this diffuse lymphoid tissue is also present. So this is another example of gut associated lymphoid tissue known as appendix. Now that is all about the microscopic features of lymphoid organs. This is just a tabulated form of comparison between the major lymphoid organs: thymus, mucosa associated lymphoid tissue, which can be appendix, which can be Peyer's patches, lymph nodes and spleen. So when we talk about cortex and medulla is distinctly present in thymus, and it depends upon the number of lobules. If there are many lobules, there will be many cortex and medulla. There is no cortex and medulla in the mucosa associated lymphoid tissue. Lymph nodes, yes, it is present, but in one lymph node, only one peripheral zone is known as cortex and central zone is known as medulla, and there is no cortex and medulla in the spleen. When we talk about the lymphatic nodules, in the thymus there is no lymphatic nodule, but in place of lymphatic nodules we have cortex and medulla appearing as lymphatic nodules, but actually they are cortex and medulla of each thymic lobule. In the mucosa associated lymphatic tissue, we have just seen that in the Peyer's patches as well as in the appendix we have lymphatic nodules present. In the lymphatic nodules are also present in the cortex or cortical area of the lymph nodes, and in a spleen it is present in the white pulp. When we talk about the lymphatic vessels, there is no afferent in the thymus because it is a primary lymphatic organ. Few efferents are present in the septa. So basically the lymphocytes are pouring into the thymus through the bloodstream. In mucosa associated lymphoid tissue also there is no afferents, only efferents are present there. In the lymph nodes we already talked about that convex part is the receiving end for the afferents, and they pour their secretions into subcapsular sinus, and subcapsular sinus will pour their secretion into the trabecular sinuses and reach to the medullary sinuses, and from medullary sinus the efferent lymphatic channels will leave the hilum. So yes, afferent and efferents are present in the lymph nodes, and efferents are only present in thymus and MALT. In spleen, no afferents are present, only the efferents are present in the trabeculae. So afferent and efferents are only the characteristic feature of lymph nodes. Unique features of thymus, as we have discussed, that in the medulla of the thymus we have Hazel or thymic corpuscles. Their function is to secrete cytokines, and also epithelial reticular cells in the cortex and medulla, which are known as Tech cells or thymic epithelial cells. We have discussed them in detail. In the mucosa associated lymphoid tissue, the crypts or the grooves are lined by surface mucosa. In the tonsils, we have discussed that tonsillar crypts are present, which increase the surface area, and they are lined by or filled with lymphatic nodules. Also contains M cells in the mucosa over the Peyer's patches in the MALT. In the lymph node, very thin paracortical region between cortex and medulla we have already discussed, and the specific feature over there is that high endothelial venules are present there. The main function of these and high endothelial venules, that the T cells which are coming from the thymus, they are pouring from these venules into the lymph nodes. So lymph nodes are filling by the T lymphocytes in the paracortical area with the help of endothelial venules, and medullary cords and sinuses are present also in the medulla of the lymph nodes. In spleen, a 20% white pulp is a component which contains the central arterioles and lymphatic nodules. In major red pulp components are the splenic cords of Billroth and sinusoids. So that is all about the microscopic structure of the lymphoid tissue. So if you have any question, you can please email. Thank you very much.